Okay, so I'm with Christoph Ammer, who published a PMAP study in gut last year. Christoph, can you tell us about what your paper brings to the community about acute pancreatitis?
So thank you very much for this question and for this interview. We were interested whether the microbiome, the gut microbiome and the oral microbiome is associated with the severity of acute pancreatitis. And we just know there might be some links because we know early enteral feeding is beneficial. We know that probiotics might be not beneficial, but there was no structured assessment of the early microbiome and pancreatitis, and that's why we just initiated the PMAPS trial. Pancreatitis microbiome is predictor of severity, is an abbreviation. And we just collected, in the very early phase of acute pancreatitis, we collected buccal swabs and rectal swabs and associate those microbiota with the outcome, with the severity, with the Atlantic classification, mortality, length for hospital stay. We needed a quite large sample size, that's why we conducted it European-wide, multicentric, 15 centers, 8 countries, and we could enroll 450 patients within 2 years. And we sequenced everything in Göttingen, so all the samples are shipped to Göttingen, and find out that indeed there's an association, not in the oral, but in the rectal microbial community, with severity, with Atlanta, etc. And that was a nice point, but the species what we have found that are most likely beneficial species that were butyrate producer, short-chain fatty acid producer, and this is quite contradicting because we always thought that are the good cops in our body, and now they are associated with the worst outcome, and that's why we just initiated the follow-up trial.
Okay, and how would you think briefly how would this impact onto clinical practice and eventually intervention?
So our main aim was to develop a biological biomarker, a diagnostic tool to predict severity. And but we agree that maybe the species and the microbiome samples that will never be in a clinical diagnosis. It's too expensive and too heterogeneous. And that's why we just thought that maybe not the species are the key or the bacteria are the key, but more this results, the pathways, the functionality of the microbiome. That's why in the PMAPS2 trial we focus more on the microbial-derived metabolites and also sample plasma and stool besides the swabs to assess the metabolites and associate them with severity, because metabolites are easily to from the blood, taken from the blood, and are easily to assess in a laboratory, and might be also cheap. That might be the good clinical marker for predicting severity, and have a biological biomarker, not only with clinical scores. So that's the aim. Let's see.
Okay, good. I guess, Philippe, you have other questions regarding your other presentation?
Yes, we will be very interested in the PMAPS2 study and its development, especially in a clinical application. You also presented a very nice study concerning PPIs and necrotizing pancreatitis and LAMS, what we call apposition stents. You demonstrated that when using PPIs, there is without PPIs there is less occlusion of stents and less necrosectomy. Is that?
Yeah. That was a totally different trial, and that was more or less a question: do we need the gastric acid to better have a better resolution of ward of necrosis?
Yes, but there maybe have a link maybe with microbiomes. There is some hypothesis?
It could be, because we know that PPIs are microbiome modulator, a strong modulator, and that's quite known. And here, but that is not the first idea we had, is more or less that we need the gastric acid which runs through the lungs into the necrosis and somehow digested, makes it more fluid, so that was the idea. And that's why we just said, okay, we retrospectively collected data from 17 centers in Europe and had almost 900 patients. And then we associate the PPI intake with, as you mentioned, first the occlusion rate and secondly the necrosectomy rates. And that was a really evident and significant association between PPI intake and both endpoints. Though it might not be beneficial to have PPIs regarding this retrospective data, with all the limitation of retrospective data, it's not beneficial to have PPIs in this kind of cohort.
Yeah, this is very interesting thing, and I think it can be a call for all French centers to participate to your study, because we observe that there is not so much French center, and we will look very interestingly at your results.
Yeah, we are very happy.
Thank you
Thank you very much.
Thank you very much.